All right, let's talk AP Biology genetics. So, there's a general idea called Mendelian inheritance. Mendelian inheritance is this idea derived from this guy named Gregory Mendel, who made a bunch of peas basically have sex, and he found out that traits are inherited from parents. 50% from mom, 50% from dad. It's not just like this idea that your hair color is just given to you by God or whatever. There is a reason to everything. So, a capital letter gene, or a capital letter allele, as you want to call it, is a dominant trait, and only one is required to express its phenotype variation. So, if we think to the real world, for example, this is a bit of a simplification, but you see a lot of brown hair and not a lot of ginger hair. So, ginger would be a recessive trait, right? A trait that two alleles are required to express its phenotype generation, while brown-haired is a dominant trait. So, if a brown-haired person and a ginger-haired person had a child, their kid would probably have brown hair. But if a ginger kid or a ginger person and another ginger person had a kid, then their kid would probably be ginger. So there is some definitions and terms that you want to know when it comes to describing these alleles. You have homozygous, which is the same allele twice, so one from mom, one from dad, both capital letters or both lowercase letters, or heterozygous, which is different alleles, so like capital A, little a, stuff like that. Let's take a look at an example that isn't with the letter A. We have the capital B is for a block-shaped animal, and a little b is for a bubble-shaped animal. As you can see, this is the dad and this is the mom. So you have something called a monohybrid cross here, and it's monohybrid because you have one cross for each one. It's a monocross in the sense that there's only one trait being shown here. So you have a capital B and a little b for this specific block-shaped animal, and two little b's for this animal. So you just move one big B here, one big B here, and one little b here, one little b here. Then you have this little b right over here. You have one little b here, one little b here, one little b here, one little b here. It's like those pictograms, if you've ever done one of those. So it's just by row. So anything in this column would have a big B, and everything in this row would have a little b, so on and so forth. So if you look at the ratio, it is from big B, little b to little b's. It's a 1-to-1 ratio, right? For each big B, little b, there's one little b. And the phenotype ratio is from block shaped to bubble shaped is also 1-to-1. So what that means is that there's a 50% chance that their kids will be block shaped and a 50% chance that they will be bubble shaped. Now, this is a simplification. It is really lucky that Gregory Mendel used peas, as most traits are just, expressed by peas by one gene. So just one gene does the color, or just one gene does the shape, or the color of the petals or whatever. But this is not always the case. For example, polygenic alleles are when multiple genes code for one trait. So let's think, for example, height. There are so many contributing factors that cause someone to be taller, right? Even if it's a small thing, like if you have an X or a Y chromosome, right? As you can see that usually, most often, people with Y chromosomes are taller than people with just two X's. Then you also have pleiotropic, which is when one gene codes for multiple alleles, right? Or multiple traits, sorry. So, for example, let's say that having a big B makes you block-shaped, but it also gives you two eyes, right? Just something like that. That's just multiple traits, just like that. Inheritance won't always follow the pattern of just one is better than the other, so it's just going to overpower it. There is something called non-Mendelian inheritance. The first one is an incomplete dominance, if a genotype is heterozygous, big A, little a, the phenotype is a blend. So, for example, if a red flower and a white flower were to breed, let's say, the child or the offspring would be pink flowers. Codominance is basically the same thing, except each trait is equally represented. So if I had a red flower and a white flower, the kid of those two flowers would be a flower with red and white petals. So it'd be like red, white, red, white, red, white, red, white. It is equally represented throughout the thing. Another way to think about it is those standard cows that you see, the white and black ones, those are, in fact, codominant cows, right? Those little spots that you see is from a black cow, and the white is from a white cow. That's how that's done. Now let's take a look at pedigrees. The pedigrees are a way of showing these traits. The squares are guys and the circles are girls. We're going to look at not recessive or dominant traits, although these could be shown on the test, but we're going to look at a specific one called X-linked recessive and X-linked dominant. So let's say we have a thing called a parent generation, where the mom and dad, the dad has an X little a and a Y, and the mom has an X big A and an X little a. So the dad has a single X little a, but he also has a Y. A Y is always treated as a recessive allele. So because he has two recessive alleles, he is showing that trait. That's why he's darkened, right? So now this X little a is expressed for this kid right here, and this X little a is expressed for this kid right here, right? They're bringing half of their traits to this kid. But for these other two kids, it's just Y's. So a Y gets brought down here and a Y gets brought down here. Now the options are, there's like a 50-50 chance at this point for the mom, right, because it's a circle, to give a big A or a little a for the X trait. So if it's X little a and a Y, it will be shaded. If it's an X big A and a Y, it will not be shaded, all right? The gene will not be expressed. It's pretty simple overall. It shouldn't be too hard to show. But here's an interesting one right here. Let's say we have a mom that is showing the trait and a dad who is not showing the trait, right? We have an X big A and a Y. There is no possible way for there to be a boy offspring that has a there's no way to have an offspring that is a boy who is not showing the trait, right? Because the dad will always give a Y and the mom will always give an X little a. But if it's a girl, there is a 100% chance that the dad will give a big A and the mom will give an X little a, right? Because the mom can only give an X little a, and if it were to be a girl, then the dad would have to give an X big A. Now let's take a look at X-linked dominant. It is almost the exact same thing, except now having a big A is what leads to the gene being expressed. So this is basically the flip of that. So from here we can see that all four of these options are possible. I would say that this is a good time to pause and take a look at what I've written here. These are all like possible things that could happen. It'll change depending on the generations and the partners that the squares and circles choose. But, that's just the way the world works, I suppose. I'd say pedigrees are pretty useful when it comes to deciphering what causes a trait to be expressed. Because if you didn't have this information, you could find out that this is X-linked recessive just based off the fact that all the guys are expressing the trait. Now let's take a look at Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium occurs in a population if there is, or is true in a population if there is a large enough population, random mating occurs, there's an equal likelihood of reproduction, there's no immigration or emigration, so that means that no animals are leaving and none are entering, and there are no mutations. And if all of that is true, the following is true. P is equal to the percent frequency of a dominant allele, and Q is equal to the percent frequency of a recessive allele. So P plus Q is equal to 1, right? So if I had 90% dominant and 10% recessive, that would equal 100%, right? So any number that is a percentage, you just divide by 100 to get its probability decimal number. So yeah, for example, 90% would be 0.9 and 10% would be 0.1. Add those together, you get 100. Or you get 1, sorry. Now you can take this a step further. P squared is equal to the percentage of homozygous dominant alleles. So that means that you have 2 big A's. 2pq is the percent of heterozygous alleles. So you have 1 dominant and 1 recessive. And q squared is the percent chance of a homogeneous recessive. And this equation right here is equal to 1. So all these added up will add up to 100%. So let's take a look at an example question that you might see. I chose a pretty hard one so you could, really understand, where to get at for these. These are not too bad once you start looking at them, but let's just go through it. So phenylketonuria is a severe form of mental due to a rare autosomal recessive allele. About 1 in 10,000 newborn Caucasians are affected. Calculate the frequency of carriers. So carriers are people who don't express the trait but do have a dominant allele there, right? So we can assume that phenylketonuria is a 0.0001 chance, or a 1 in 10,000, right? That's q squared, because you have to have both recessive alleles for that to be the case. But this is q squared. We need to find q in order to find p. And how would we do this? Well, we square root it. You got 0.0001 square rooted, you get 0.1. Now all we have to do is plug that into this equation. You have p plus 0.01 is equal to 1. So by that logic, you can find out that p is equal to 0.99. Now if you were to square 0.99, you'd get 0.9801. Now, if you go through this equation right here, the one that we had from the beginning, and you just start filling in the blanks, right, substituting it in, so you have 0.981, you bring that over here, you have 0.01, you bring that over here, you can calculate that 2pq is equal to 0.1899. Or another way you could do it is you could multiply p, right, which we found as 0.99, and q, which is 0.01, and then multiply that by 2, you would get 0.0198. Now, leaving it just like this is good enough. Do not divide from here. Do not go, oh, I just want PQ. You do want 2PQ. The chance of someone being a carrier, a Caucasian person to be a carrier, is a 0.1899 probability, or basically a 19% chance, or sorry, a 1.9% chance, or I rounded it up here to 2% chance that a Caucasian is a carrier, not a character, a carrier of phenylketonuria. Now, Hardy-Weinberg equilibrium is not the only kind of equations you can expect to find on a genetics test. You'll also find chi-squared. Chi-squared is this thing right here. But it's also a statistical test used to determine if the data we collect is significantly different from what was expected. So let's look at an example problem. I'm at a gambling parlor and I bet a lot on black and it ended up being green. I suspected that there was some cheating afoot, so I took some observations of the roulette table. So I looked at 100 rolls of the roulette wheel, and I found out that there was 58 times when it landed on red, 32 that it landed on black, and 10 where it landed on green. So this all adds up to 100. But what we should expect is that there is a 49 rolls landed on red, 49 landed on black, and 2 landed on green. Because if you look at a roulette wheel, the only 2 that are green are 0, and 0, 0. And the rest are just red or black, right? So that's why these two are even. And the roulette wheel is out of 100. I don't know if you're not like a big time gambling fan. So to find chi-squared, you have to do observed minus expected squared over the expected value. So I'm just, ignoring the work that I might have done for this, but it would have been 58 minus 49 squared over 49, and that would have got me 1.65. The next one would have got me 5.9. And interestingly, the green one would have got me 32. So my sum chi-squared number is 39.55. Now, my null hypothesis is that there was no cheating at all, right? Green is equal to 0.02, and the red and black are the same. So that would actually look like that. And green is equal to 0.02, and this would say that red and black, and that's not equal to 0.02. My alternative hypothesis would be that red and black are not the same, and green is not equal to 0.02 probability. Now, there's a thing called a critical value, which I'll explain in a second. But what you need to know is that if chi-squared is less than or equal to the critical value, then you fail to reject the null hypothesis. That would mean that if my number was lower, then that roulette table is just, doing what it should be on a random chance. It makes sense for it to be randomly doing that. But if chi-squared is greater than the critical value, then there is something fishy afoot, and you can definitely say, I think that you are cheating. So critical value is based off the alpha level. In this case, let's say that we're 95% sure, so our alpha level is 0.05. And the degrees of freedom. To find the degrees of freedom, you have the number of categories, we have 3, and we subtract it by 1. So we get 2 for this case scenario. You will find a table on whatever test you're taking where you will find the degrees of freedom and the degrees of freedom and the alpha level, and you'll put those together and you will get a number, right? For example, for me it was 5.99. That is your critical value. So now if we compare it to our chi-squared, 5.99 is less than 39.55. So we can confidently say that we reject the null hypothesis, and we are 95% confident that that person at the roulette table who was rolling it is a cheat, and he stole all my money maliciously. It was not just random chance, or at least we're 95% sure of that. Anyway, that's all that you need to know for genetics. You will not get a Las Vegas type question when it comes to chi-squared. You'll probably get something based off I observed that 30 people out of 100 are blonde-haired when I expected to see only 10. Is there something fishy going on? Right? That's what that would look like. But I thought I would do something more simple to understand with basic numbers like 49, 49, and 2. Anyways, all that being said, that's just about everything that you need to know for genetics. I'll add something very brief into here. There is 2 kinds of chromosomes. You might remember this from the chromosomes unit, but anything that is not an X-linked trait or a Y-linked trait or something like that is an autosome chromosome, right? So that means that it is not based off sex, right? But a sexed chromosome is just your X or Y's. So if you have a sex chromosome, that's X or Y. If you do not have an X or Y chromosome that's being focused on, then it is an autosome, and that means that it's not one of these. Another one that I failed to mention here is a Y-linked trait, but that one's pretty simple. If a kid has a Y and they have that trait, well, then they're going to have it. It's that simple. Anyways, that is actually it. Good luck on your test, and yeah, have a good one.